Hey guys, so this is our week 15, week 16 wrap up. So we are, it's our last wrap up video and we are wrapping up the semester. As a reminder, you guys got an email about a SETI, which is a student evaluation of teaching effectiveness. Please fill those out. Constructive comments are appreciated, especially since this is the first time this course has been taught asynchronously. And I want to thank you all for kind of working with the bumps, the ups and downs, of this semester, trying all the new technology, new online platforms. I really appreciate your patience and your feedback. And again, just reminders, letting me know when something goes awry. Couple reminders, there are 2 cellular respiration Lapser simulations. You only have to complete 1. So if you've already done it, then you do not have to do the second one. I'm going to combine all those scores. If you happen to do both, I'll take the higher score. And so this is a little test I'm doing with Lapser, but if you've already did the first version, then that's fine. No need to do it again. Okay, and also your final is this week. I know on the syllabus it says the date of December 7th because it's just for this week, but like all our previous midterms, the final will open up this Thursday at 12 a.m. So right at the start of the day, and it'll be open all day Thursday and all day Friday. So it'll close at 11:59 p.m. on Friday night, and you'll have 2 hours this time to take the test, unless you have other accommodations. All right, you guys, so let's get into this last section. Quick note, as we get into the biochem, it can get kind of complicated. I put a lot of sections on there just for review. You can tell if it's just for review because I didn't put any in lecture questions. And so if a section has no in lecture questions, then that's just for review, kind of for your information only. And looking at some of your comments on the discussion board, it seems like some of you appreciated that. So I'm glad I put it in there. But yeah, so I don't plan on assessing you on those sections. Okay, so one section you guys thought was really hard, which it is, is the properties of amino acids. And so I replaced the figure that was originally in the top hat content. So I changed it to this one, which is a different one. It's okay. But make sure when you guys are looking at the properties of amino acids, you have to look at the R group. Remember, every amino acid, as you can see here, has a carboxylic acid. These all have carboxylic acids, every single one. It's kind of the definition of an amino acid. They all have this amino group as well. Okay, this one's a kind of special case there. They all have that amino group. And so what makes them special are these R groups, okay? These R groups are what makes them special, okay? So if the R group is mainly carbons and hydrogens like these ones, then it's considered nonpolar or what we consider hydrophobic. It would be found in hydrophobic environments, which are usually the interior of proteins, because the inside of our body has a lot of water. It tends to be an aqueous environment. So the amino acids that are on the outside of a protein are the more polar ones to interact with the water. So these ones, the non-polar hydrophobic ones, tend to be kind of they huddle together in the interior of proteins in the core. Okay. See these ones. Let's go down to acidic. The acidic ones all have a carboxylic acid group. Okay. So remember, all amino acids have carboxylic acids, but in the R group, if it's a carboxylic acid, then it's considered acidic. Now, the basic ones, they have nitrogen in them, okay, these amino groups. And remember, in the R group, it has that amino group. But there's these 2 over here, glutamine and asparagine, that also have that amino group in the R group. But notice that these ones are next to a carbonyl. They're next to the C double bond O. And these ones are considered polar neutral. So they're not basic. Remember, a base is something that can accept a proton. These ones, lysine, arginine, and histidine, can accept a proton, okay? And remember, acids, acidic, they're acidic because they can donate a proton, which is something that we learned about carboxylic acids. That was one of the properties of carboxylic acids in our OChem section, and acids in general in our Gen Chem section, that acids donate protons. See, what a great question for a final, you know, that incorporates Gen Chem, OChem, and Biochem, right? We learned about acids, we learned about carboxylic acids, now we're learning about amino acids, okay? Ooh. All right, and so the last category here are these ones that are polar but neutral, so they're not acidic and they're not basic. And they have these R groups that have like OH groups in them or amino groups in them, OH, a thiol group, SH, these carbonyls. So they're not just carbons and hydrogens, okay? So that's what I want you to gather from the properties of the amino acid section. We have to look at the R groups. And again, using principles that we've learned from OChem and Gen Chem, we can classify them as either nonpolar, which would be found in hydrophobic environments, or polar, and these three are all polar. The basic, acidic, and polar neutral are all considered polar. It just happens that some of them are also can act as acids, so they're acidic, meaning they donate protons, and some can also act as bases, so they're considered basic, meaning they can accept the proton. And then these ones don't either, but they can typically hydrogen bond, and so that is a polar neutral, okay? Remember, you have to look at the R group. Okay, clear my drawings. All right. Okay, so this was a question, and I apologize. Some of you came to office hours and talked to me about this, but the strongest interactions between peptide chains are, so these are your options: London dispersion forces, disulfide bridges, hydrogen bonding, dipole interactions, and ionic interactions. I had a mistake, so as long as you guys all put in an answer, I changed it to 100% participation. And I even gave the wrong answer in office hours. I apologize, but it should be disulfide bridges. And the reason is because a disulfide bridge is a covalent bond. I mean, it's actually sharing electrons, okay? And so the disulfide bridge is pretty strong. Right, types of interactions. So again, these are the different types of interactions, that we've seen. So we can have hydrogen bonding when we have a hydrogen bonded to an oxygen and nitrogen or fluorine, and it can create this interaction we call hydrogen bonding. There's ionic interactions where we have a negatively charged atom interacting with a positively charged atom. And then we have these hydrophobic interactions, which have carbon hydrogen groups that are nonpolar, right? They're nonpolar and they tend to cluster together and they form these hydrophobic pockets, which we consider hydrophobic interactions. And then there's these disulfide bridges, which are when you have something like cysteine that has a thiol group. Remember, it can get oxidized and form this disulfide bridge. Again, we saw this in our organic chemistry section when we were looking at thiols. Two thiols can be oxidized to form a disulfide. They can also be reduced to get back to those two thiols, okay? All right, enzyme action. So this is one of the questions you guys were confused about, but what type of interaction would occur between an NH3 group present on a substrate molecule and a functional group of the active site in an enzyme? So you just got to think, if you got something positively charged, like this NH3 group, then remember it's attached, right? So it has a fourth bond on it. That's why it's positively charged. Then it's going to probably interact with something negatively charged on the active site of the enzyme, okay? And we would consider that an ionic interaction. Okay, so then we got into mutations and DNA and RNA. And so this was one of the questions you guys had some questions on. Actually, you guys are okay with this question. It was the one after this, but I want to start here. So it says a portion of a coding strand of a gene was found to have the sequence this. A mutation occurred in the gene making the sequence this. Identify the mutation as substitution, an insertion, or deletion, okay? And so if we go through, and I lined them up here, if we go through and look and see, here's a change. So this T got switched to a C, but everything else is the same, okay? So we have a T to a C. So we did a substitution. We took one out, we took the T out, and we substituted a C in, okay? We didn't insert an additional thing, and we didn't delete, okay? And let's take a look at what would happen. So this is a substitution. But the second question, the follow-up here is, what effect would this mutation have on the amino acid sequence of the protein obtained from this mutated gene? And you have to use the genetic code to figure this out. But real quick, let's look at the different types of mutations. So mutations alter the mRNA, and therefore they can alter the proteins. So remember, that was the coding strand template. From the template strand, you can get the mRNA, right? And so A matches with U, right? T with A, G with C, C with G. And we look at this and we see what the mRNA would be. It'd be UUU. And the protein that codes for UUU is phenylalanine, okay? And that's kind of the going from DNA to RNA is the transcription, and then going from RNA to protein is translation. Now, if we just do a substitution, like if we change the CGG to CAG, well, instead of getting alanine here in our protein, when we go through translation, we'll get valine, because GCC codes for alanine, but GUC codes for valine. So, but then the next protein, it picks right back up, right? So the only one that got affected was this one amino acid, but the ones before it and the ones after it are not affected. So it's just a single mutation in the amino acid sequence. Now, what happens with an insertion or a deletion? Okay, these are also considered what we call frame shift mutations. This is right. So if instead we inserted an additional C at that point, then everything would get shifted down, right? And now everything that came before it is still the same, but then everything after an insertion is going to change because you're shifting that frame, right? So instead of being this CGG, it's now CCG. So you have a C here. And then the next one is going to be GTC, which is here, GTC, right? Everything gets shifted down. So all the amino acids that come after that insertion are now different. Big difference, right? Same idea, something's deleted, a deletion, or removed, right? If you remove a protein, then you're shifting everything, by one less. And again, you get this whole shift, and all the proteins, all the amino acids, in the protein are different. So that can also have huge effects on your amino acid sequence, okay? So if we go back and look at that question, here's a little simpler table than the one you have on your top hat, the genetic code. Here is the DNA. Here's the RNA. So I just changed all the T's to U's, okay? And if I look and see what would happen, right, I would see, let me see, right, I have AUG here, and I can look, AUG is methionine, that's the start. Then I have AGC, and AGC is serine here. Then I have GAC. GAC is here, okay, aspartame, okay. UUU, phenylalanine, okay. And that's where our change occurs, because we have AUG, then we have AGC, which is the same up there, and then we have GAC. But instead of UUU, we get CUU. So remember, UUU is for phenylalanine. CUU is for leucine. Okay, but then after that, it gets back to normal. Now we're at CGC. This is CGC, CCA, CCA, UUA, UUA. So here with just the substitution, the effect is just we go from a phenylalanine to a leucine. Okay. All right. And then the last little point, a lot of you weren't asking questions on this, but with like the citric acid cycle, I did change the picture on that one too because I found some errors with the picture I had in there. So you guys should be able, and I know this is probably going to be one of the more difficult things I ask you guys to do, but can you look at these reactions and identify what's going on? So imagine you had a figure like this, but without these gray boxes explaining what's going on. um, but like if you see a water molecule leaving here, notice, right, you had a water molecule. It removed H2O, an OH from this carbon and a hydrogen. And now there's a double bond in there. So it's a dehydration. And we've seen that in organic chemistry. The dehydration of alcohols formed alkenes. And look, alkene double bond. Okay. So see that? Ooh, that's really cool. Again, the single double bonds, that's from our Gen Chem section. The dehydration of alcohol to form an alkene, that's from our OChem section, but now we're looking at it in the context of citric acid cycle, biochem question. So one question can incorporate all three. That's so cool. Okay, and then look, then they're adding water back in and you get the alcohol back. So hydration of alkene, we saw that as well, okay? Here we're removing a carbon. So just be cognizant that, you know, here we have 1, 2, 3, 4, 5, 6 carbons in this isocitrate molecule. We have this arrow showing CO2 leaving. We removed a carbon. So we have 1, 2, 3, 4, 5 carbons left. Then we lose another one. Okay. If we keep going through, whenever you see the word dehydrogenase, I don't plan on testing you on this, but when you do see dehydrogenase, well, I do plan on testing you on this, but you guys should be able to figure out dehydrogen, removal of hydrogen, right? It's removing hydrogen. And we talked about when we remove hydrogen, that is a reduction. Oh, that's an oxidation, okay? Something is getting oxidized and something else is getting reduced. So the FADH2 is what got reduced. Our substrate succinate here is what got oxidized, okay? In this next step, we have an alkene. There's a double bond here. We hydrate it, we add an alcohol, and guess what? There's an alcohol, okay? And so we've seen that before. Hydration of alkene gives us the alcohol, okay? Then we have another dehydrogenase. We remove hydrogen, okay? And therefore we oxidized our malate substrate, okay? So kind of being able to look at molecules and see what's happening. I don't expect you to memorize all these reactions, but I'll probably ask you about the types of reactions that are happening. Okay, I think that's it. Yep, that's it. I wish you guys all luck on your final. I hope you have some productive studying going on with this final and all your other finals you go on. I hope you guys are all safe and healthy and taking good care of yourselves. All right. Please email me if you have any questions and have a good break.